Hello, everybody. I'd like to start thanking Sander Wezenberg for giving me the opportunity to present my work in this focus session on the Change conference. And I'll tell you about our research using macrocyclic receptors for the transmembrane transport of anions. What I'll do today, I'll first introduce the topic of transmembrane transport, and I'll explain how we can use synthetic carriers to do this. And then I'll take you on a journey through 4 examples involving the transport of propylammonium chloride ion pairs by Calix 6 arenes. Then we'll move on to bambus uril macrocycles for the transport of chloride and bicarbonate. I'll explain about an assay we developed to study bicarbonate, and then we'll return to Calix arenes, now Calix 4 arenes for the transport of copper cations. But first of all, why are we interested in studying the transmembrane transport of anions? It's because this is such an important process in biology. So cells have exterior membranes. Also, the compartments inside the cell are all separated by membranes, and these membranes are impermeable to most ions. So that's why cells have proteins in their membrane that take care of their transport. But when there's a problem with this transport process, for instance, due to the genetic mutation of such a protein, this can give rise to all kinds of diseases. The most well-known example is probably cystic fibrosis, which is caused by deficiency in chloride transport. So a chloride transporting protein is not doing its job. So what we are trying to do is using synthetic molecules to take over the role of these proteins. So these are molecules embedded in the bilayer that extract the anion, take it through the membrane to release it on the other side. And here in a video, I'll show you how this works. So here we have ions on one side of the membrane. They try to get through, but they are too hydrophilic to penetrate the apolar interior of the lipid bilayer membrane. But now if we have synthetic ion carriers that can bind these anions and shield their charge from the hydrophobic interior of the membrane, they can carry them through to release them on the other side. Well, and these carriers, these hydrophobic carriers, stay inside the membrane so that they can go back and forward, taking more and more ions through, and some of that are very efficient in doing so. So what we need in a carrier in order to do this is on one hand a binding site, and on the other hand a lipophilic scaffold that helps to make sure that it's well embedded in the lipid bilayer membrane. Well, here are some examples of structures that were found to be efficient chloride transporters. And you see in all cases we have such hydrophobic scaffold, for instance, these decalins or an anthracene, and we have groups that can bind, hydrogen bond donor groups to bind the chloride anion. Here in most of these examples for efficient chloride transporters, these are ureas or thioureas or pyrulic NH-based hydrogen bond donors that bind the chloride anion. Well, then the question is, how can we study the transport by such carriers? And for that we use liposomes as model membranes, and in these liposomes, in the membranes, we can incorporate our synthetic molecule that functions as carrier. Then we see here the fluorescent dye lucigenin that is encapsulated, and this dye lucigenin, its fluorescence gets quenched upon increasing concentrations of chloride. So we can add sodium chloride to the exterior of these liposomes to create a chloride gradient. And then if the carrier is active, it can take chloride into the liposomes. However, then we will build up negative charge inside. So we need another anion that can be taken out of the liposome to balance the charge. That's why we use sodium nitrate as a default for such experiments. And this process, this chloride transport process, we can then monitor with fluorescent spectroscopy, where we see, after the addition of the chloride, the quenching of the fluorescence of lucigenin, indicating that transport indeed happens. And at the end of the experiment, we could add some detergent to lyse the vesicles to verify that indeed transport took place and for normalization purposes. Okay, this brings me to the first example, the anion transport by Calix 6 arenes. And most of this experimental work has been done by PhD student Glenn Grauwels, well, now a doctor, Glenn Grauwels, in close collaboration with my direct colleague Yvan Jabin and Christine Bartik. So Glenn studied a series of 4 Calix arenes with urea or thiourea groups in this lucigenin assay that I just explained. And what we see is that the compounds that just have phenol groups did not show much activity, but when we had CF3 groups present, there is activity, and the thiourea is more active than the urea. So that's a nice result, is to show that Calix 6 arenes can be used for the transport of ions. However, not so interesting, because this Calix arene, if we just use the hydrogen bond, the thiourea hydrogen bond donor groups, it's not so different from all the examples I showed a few slides back. But Calix 6 arenes have an interesting feature. They also have this macrocyclic cavity. And this macrocyclic cavity is well known to be a good binding site for propylammonium cations that fit in very nicely, forming actually an ion pair with the anion bound by the thiourea groups. So what we tried is to transport propylammonium ion pairs. So for that we used again this lucigenin assay, but now replacing nitrate by sulfate. Well, what happens if we then add sodium chloride to the outside? We see a tiny bit of transport, some chloride goes in, but then transport stops very quickly because sulfate is too hydrophilic to come out. But when instead of sodium chloride, we added propylammonium chloride, we saw fast transport indeed. And alternatively, we could first add the chloride and then the propylammonium cations to get this transport. Well, to fully prove that we have indeed propylammonium chloride going inside, Glenn used NMR experiments looking at what ended up inside the liposomes, and this confirmed indeed that propylammonium chloride is transported by this Calix 6 arene. So this Calix 6 arene uses its macrocyclic cavity to bind the propylammonium, while the chloride binds to the thiourea groups, and then the ion pair is taken across and released on the other side. So this is the first example I wanted to show you. This brings me to the second, which is about Bambus uril macrocycles. These Bambus uril macrocycles were developed by Vladimir Sindelaar and his group from Masaryk University in Brno, Czech Republic. And these macrocycles have a very special property. So they have 12 hydrogen atoms, methane hydrogens pointing into this cavity. Well, and upon addition of more electron withdrawing fluorine atoms along this series, we see that the cavity in these images gets more blue, which means the cavity gets more electropositive. And that leads to very high affinity constants. Here for compound 4, we have 10 to the power 11 for nitrate, 10 to the power 10 for chloride. That's ridiculously high affinity constants. And I should remark that here we do not have acidic and H atoms. It's just 12 polarized CHs inside this cavity that work together to create this very strong anion receptors. So we've tested these for transport in the standard lucigenin assay looking for chloride-nitrate exchange. And the result was a bit disappointing. Only compound 4 showed transport, but here we had to go to relatively high concentrations of 1 transporter per 200 lipid molecules. However, when we changed the conditions a bit and we replaced the nitrate by bicarbonate, we found much better rates of transport. Here, the transport by compound 4 is very fast, and that while the concentration is 25-fold lower than in nitrate. So here we now have very efficient chloride bicarbonate exchange by these bambus urils. Well, why did it then not work in nitrate? Well, our hypothesis was that the binding affinity for nitrate was too high, preventing the bambus urils from releasing this anion. We've tested this. This green curve shows transport of chloride in bicarbonate solution, so chloride bicarbonate exchange, and then upon addition of 10 millimolar or even only 1.5 millimolar of sodium nitrate, we see that the transport is brought to a halt. So a bit of nitrate stops the exchange of chloride and bicarbonate, indicating that nitrate is bound too strongly for efficient transport. But it doesn't answer the question why chloride bicarbonate exchange is so fast, faster than any other compound reported in the literature. So to understand that, we got some interesting information from binding data. Because the NMR titrations indicated that the bambus uril could not just bind a single chloride or a single bicarbonate anion, but it could also bind the pair of these ions simultaneously. Here we see the chloride in green, here we see a bicarbonate, and there is a hydrogen bond between these two anions inside the bambus uril. Well, that's a bit similar by what Amar Flatt has found for other types of anion receptors for hydrogen sulfate. And we think that this complex could explain why transport is so fast. Because in the case of nitrate, chloride nitrate exchange, nitrate has to be released to get the empty bambus uril before the chloride is bound, then chloride is released before nitrate is bound. But once we can form this binary complex, we can first bind the chloride before releasing the bicarbonate. And this second affinity constant is much lower, so this would be a much more favorable exchange process, which is probably quite a bit faster. So we think this could be why the bambus urils are so efficient chloride bicarbonate exchanges. So I've told you in this second part why bambus uril are good transporters for chloride and bicarbonate. However, we've been only looking at the transport of chloride into the liposomes. We could not look at transport of bicarbonate because there was no assay, no method around to study the transport of bicarbonate directly. And we were very keen to confirm this result by monitoring bicarbonate. So then at a conference, I met Steve Butler from Loughborough University in the UK, who had developed this europium complex. And this europium complex can bind bicarbonate, and upon binding bicarbonate, the different emission bands of this europium complex increase. So I've encapsulated this dye in liposomes, added some bicarbonate, and yes, over time we saw this increase of the excitation, which is plotted here as emission over time. Addition of bicarbonate, we indeed see the increase of the transport. So this europium complex can be used to monitor transport of bicarbonate in liposomes. Well, then I was lucky that Luis Martinez joined our group as a postdoc, and he has taken over this project, and he changed, for instance, the concentration of the bambus uril in the liposome, and we see that more of the bambus uril is present in the liposome membrane, the faster the rate of transport, a good confirmation that it works. However, there is a challenge with bicarbonate, and that is the fact that in aqueous solution, it's always present in equilibrium with CO2 and carbonic acids. So when we add a pulse of sodium bicarbonate to the exterior of liposomes, we also give rise to a gradient of CO2, and CO2 is known to diffuse spontaneously across the membrane of liposomes. So when we added the cation transporter monensin, which can basically exchange sodium and protons, we indeed saw transport of bicarbonate in this assay. But in this case, it's not the bicarbonate anion that is transported, it's CO2 that goes into the liposomes that would acidify the liposomes, but the monensin can get rid of the protons, transporting protons out of the liposomes to prevent this acidification so that carbon dioxide diffusion can continue and the net concentration of bicarbonate increases over time. Well, to verify that this is indeed what takes place when we add monensin to liposomes, we replaced the europium complex by the common dye HPTS, which is a pH-sensitive probe. And if there's no transporters present and we add our bicarbonate pulse, we indeed see acidification of the interior due to CO2 diffusion. However, if there's monensin present, we see a very fast equilibration of the pH gradient, because upon addition of the sodium bicarbonate, we increase the exterior pH from 7 to about 7.4, but this gradient is quickly equilibrated by monensin. So one more important thing to realize here is that this pH equilibration is very fast, whereas the increase of the bicarbonate concentration inside the liposomes is much slower, and that is because the CO2 diffusion is rate limiting in this process. Okay, but if this CO2 diffusion mechanism can play a role when we add monensin as a transporter, how do we know what happens when we have anion transporters present? Are anion transporters indeed exchanging bicarbonate anions for other anions such as chloride, or could anion transporters act as HCl transporters, facilitating the diffusion of CO2 instead of actually transporting bicarbonate? Well, for bambus urils, we had already done other studies to show that bambus urils cannot transport protons or hydroxide. Bambus urils cannot dissipate pH gradient and thus have to act via mechanism A. That's what we further tested by combining the bambus uril with monensin. And when combining the two, we see indeed a faster rate of transport, shown here in green. So with the monensin added, transport is faster than by the bambus uril alone. And that's because the bambus uril acts via mechanism A, the monensin via mechanism B. Those mechanisms are independent and we get faster rate of transport. But now when we used this urea compound and we compare the blue and the green curve, we see that the rate of transport doesn't change upon the addition of monensin. And that indicates that the urea acts primarily via mechanism C. So this HCl transport combined with CO2 diffusion. If then CO2 diffusion is rate limiting, adding monensin, facilitating mechanism B, but still CO2 diffusion being rate limiting, we see no change in the rate of transport. The good news is, however, that the transport by just the urea is still a bit faster than by monensin alone. If we add more monensin to the vesicles, we don't see faster transport because CO2 diffusion remains rate limiting. But when we added more of the urea, we saw a bit faster transport. So in the case of this urea, the primary mechanism is mechanism C, but once we get to the limit of that, mechanism A can contribute as well. Well, this is a very important result because most of the reported ion bicarbonate transporters have this acidic urea or thiourea groups and could potentially just dissipate pH gradients, facilitating CO2 diffusion instead of transporting bicarbonate. So this new assay that we have developed does allow to monitor the bicarbonate transport directly, and it allows to distinguish the actual transport of bicarbonate, for instance by the bambus uril, from CO2 diffusion-based mechanisms by cation transporters or HCl transporters. This brings me to the fourth part of this presentation, where we return to Calix arenes, now Calix 4 arenes. And these Calix 4 arenes with 2 imidazole groups can bind copper ions. This structure was already reported about 20 years ago by Olivier Reynaud's group. And the binding site very much resembles that as it's found in proteins, where 2 histidine groups can give this linear coordination of copper 1 cations. However, nobody had ever shown that copper 1 cations could be transported by synthetic molecules. So this binding was known, but Nathan set out and synthesized some derivatives with longer R groups to lock this Calix arene in its cone conformation. And he had to develop the methodology to see if these compounds could indeed transport copper 1 cations across membranes. For that, he encapsulated the dye BCS. This is known to form a 2-to-1 complex with copper cations, and that decreases the fluorescence upon binding. So indeed, when he then studied the transport by his compounds for these Calix arenes with different R groups, he saw transport, and the propyl and the pentyl group were more active than the derivative with just a methyl group that has a much more flexible geometry. And in contrast, if we only had a Calix arene or only an imidazole group, then there was no transport. So these 2 imidazole groups nicely pre-organized on this Calix arene macrocycle give indeed copper 1 transport. And this is the very first time that copper 1 transport was demonstrated by synthetic compounds. And it's quite interesting because copper in biology is quite important, and it's transported across membranes in its copper 1 oxidation state. Well, here I've shown you 4 examples of transport of ion pairs, anions or cations by macrocyclic carriers. That brings me to the acknowledgements. I'd like to thank the EMNS laboratory. Here are again Glenn, Louise and Nathan, who did most of the work I showed today. This was in times we could still actually travel together to a conference and have a meal together. I'd also like to thank my international collaborators that are listed here, and the FNRS and the ERC for funding of this work. And if you're looking for a postdoc position and you're interested in this type of research, then feel free to get in touch. We have a postdoc position open at the moment, and there's also opportunities for PhD students. Thank you very much, and I'm happy to answer any questions.